Alright, now that we know what local adaptation is, let's talk about some experimental approaches you can take to identify if local adaptation is actually occurring in different populations. And we'll talk about two different general ways to do this using a common garden, or I'll abbreviate it as CG, or a reciprocal transplant, and I'll abbreviate that as RT. So let's start with common garden. A common garden approach takes individuals from different populations in the wild and brings them into the lab and holds them under identical conditions. And so by identical conditions, we mean identical temperatures, identical salinities, identical light cycles, light source, food, anything you can think of, densities, same number of individuals in those tanks. You want to keep everything exactly the same. So this is an example when I was conducting my studies during my PhD. These are the actual tanks I used, where I would take individuals from Northern versus Southern California and bring them into the lab to have this common garden period. Normally this common garden period lasts at least 3 weeks. Ideally, what you would do is actually take these individuals from the field, bring them into the lab, let them reproduce, and then use their offspring or even those offspring's offspring in your experiments. But in general, you want to do this common garden period for at least 3 weeks. And why are you doing this common garden? So what this common garden period does is it erases any past differences in environmental history, so you can be confident that any differences you see in your experiment are in fact due to these genetic components of local adaptation. So to illustrate this point, here's a graph of temperatures, so that's the y-axis, occurring across different days at the different geographic sites that I study for my research. So again, the northern sites are the blue lines, the Southern California sites are the red shade lines. And if we look at, say, this peak around here, for instance, this Southern California site reached 44 degrees C, which is super hot, whereas that same day, the individuals in Northern California reached 30-ish, so 14-degree difference. If you took those individuals directly from the field and tested their thermal tolerance, it's very likely the Southern California population would do better just because they've already been exposed to these warm temperatures. So they've already probably made the proteins that are needed to help them survive. So that wouldn't indicate local adaptation if you took them directly from the field, exposed them to warm temperatures, and then saw which population had higher survival. What you need to do is this common garden period where it essentially gives you a blank slate and levels the playing field. So you know that every individual that you're testing in your experiment is starting with the same proteins, the same enzymes in their system already, because they've been exposed to these identical temperature and everything else conditions in the lab. Food availability is another big one. Food availability often has a large effect on an organism's thermal tolerance. So if you take individuals directly from the field, you don't know if one population has just been getting consistently more kelp, more food than the other one. So this common garden period first lets you get rid of any of these potential confounding factors. And then if you have this common garden period and still see differences in thermal tolerance, even after you've kept them in these identical conditions, then we know that the differences we're seeing are because the populations have evolved differently and selection has caused this local adaptation. So after you do this common garden period, then you would perform an experiment and determine whether your populations show different phenotypes. Again, phenotypes are these observable physical characteristics. They're kind of gross metrics, like survival is one that's commonly used. So you can just determine whether individuals live or die. For both the approaches we're going to talk about in this video, the common garden or the reciprocal transplant, both of those types of experiments are looking at phenotypes. So say you've done a common garden period, one experiment you might do then is expose individuals from those different populations to different levels of thermal stress. So this is again some data from my research where we have the percent survival on the y-axis. So here that phenotype that I was measuring is survival. The x-axis is the temperature. So I exposed these individuals to different maximum temperatures based on that graph I showed you before, based on some actual temperatures that they experience in the field. And then what you're doing is seeing if individuals from Northern California populations, if their survival differs from the individuals from Southern California. And if you look here at this graph, there are several temperatures where indeed the overall survival of the Southern California populations is significantly higher than the Northern California ones. So in other words, the survival of those red lines at each of those different temperature data points is higher than for the Northern California ones. Because we did this after a common garden period, we can then assume that these thermal tolerance survival differences we're seeing in these populations is due to local adaptation. However, there is a disadvantage to this common garden approach. There are pros and cons. So the pro is that you're in the lab conditions able to control for these other potential confounding factors, right? So you're able to make sure the only difference in your experiment is temperature. But that can also be a potential disadvantage because you're stripping out any of these other factors that might be playing a role in nature, right? So in other words, the lab doesn't equal the field. So for example, these are some snails in a tide pool in, I think, near Laguna Beach. You can see that there's some different species that are there. So when you're doing a common garden approach, you're taking out these other factors that are present in nature. One way to try to get around that is to do the other type of approach, this reciprocal transplant approach, where you can take into account more realistic scenarios encompassing all factors that are present in the environment. So in this reciprocal transplant or RT approach, what you're doing is transferring individuals between source habitats. This asterisk is here because ideally you'd actually take individuals from the field, bring them into the lab, let them reproduce, and then put the offspring back into these different sites in the field. But what you're doing is taking individuals, putting them out in the field, and then quantifying their fitness at these different locations. So that's relatively easy to do with sessile organisms like plants or corals, which are shown here. And in general, when you're doing these types of reciprocal transplant approaches, if you see something similar to the pattern we introduced when I was defining local adaptation, so if you see the performance of local genotypes exceeding that of foreign genotypes, then local adaptation is occurring. So again, this is that same figure we showed before. So here the local genotype of genotype 1 is doing better than the foreign genotype in this particular habitat. So if you saw this in a reciprocal transplant experiment, you would conclude that local adaptation is indeed occurring. So let's go back to that example I showed earlier with the snails from my research. I actually did a common garden approach, but if I wanted to take a reciprocal transplant approach, how would I do that? If I wanted to use the same populations, but instead of using a common garden approach, use a reciprocal transplant approach, how would I do that? I'll give you some time to think about that. Maybe play some Jeopardy music in my own head. I'll wait here. You can pause the video if you're still taking notes or thinking through it. I'm going to go on to the next slide. Okay, so if I wanted to take a reciprocal transplant approach, what would I do? So these are those same 6 sites that I've been focusing on, 3 in Northern California, 3 in Southern California. If I wanted to take a reciprocal transplant approach, again, ideally I would take these individuals into the lab, let them reproduce, and then put their offspring back out into the field. For these particular snails, it takes them about 3 years to reach sexual maturity, so I don't do that. I just use the adults I collect from the field. So what you would do with a reciprocal transplant approach, for some of these individuals, you would move them back into sites in their local habitat. That's to account for any handling effects or anything like that. For some of them, you would also move them to Southern California sites. And you would do the same thing for these Southern California sites. Some of them you would move back into their original local habitats, and some of them you would swap and move into Northern California. This is a little bit more logistically challenging in the field. You'd have to put them in cages, you'd have to tag them to make sure you could keep track of them. I personally didn't do a reciprocal transplant because of these logistics. I've actually done several experiments in the field with mussels, for instance, where some birds, oyster catchers, ate a bunch of our experiment. So I have a bone to pick with oyster catchers, but this is one of the reasons why I chose a common garden approach instead of a reciprocal transplant one. But okay, say you want to do reciprocal transplant, you would transfer some individuals from northern to southern sites and vice versa. And then what would the results of the reciprocal transplant look like? So assuming the same phenomenon is happening, so based on those common garden results we got earlier, is local adaptation occurring or not? Think about that, and then think about, in this scenario, in this reciprocal transplant context, what would the results look like? Again, I'll let you think about that. All right, so if you think back to the common garden phenotype survival results, we did see differences in survival even after that common garden period, which indicates local adaptation is indeed occurring. So the results we would find in our reciprocal transplant experiment would reflect that. So if we wanted to recreate this graph shown here, on the y-axis we would have some measure of fitness. We would maybe use percent survival, which is what I used in that previous common garden graph. And then we have two different environments. We would have environment 1 would be Northern California, environment 2 would be Southern California. And if we look at the fitness and survival of these different populations, we would see something like this, where for the Northern California individuals, they would have higher survival in the Northern California site, and it would drop off in Southern California, and we would see the opposite pattern for the Southern California individuals, where they would have higher survival in their native environment, Southern California, but the opposite would be true in Northern California. So this, if we retain these results following a reciprocal transplant experiment, we would again conclude that local adaptation is occurring. All right. So for both of these types of approaches, common garden or reciprocal transplant, again, we're looking at phenotypes. So the phenotypes that are a result of the different genotypes. Because again, local adaptation is caused by these genetic differences, but you can look at that at the phenotype level. And so when you're thinking about what you actually want to measure in these types of experiments, the different phenotypes, or they're often called response variables, there are a variety of different things you can measure to actually quantify fitness. So that fitness is that y-axis of that graph we've been showing. So there are several different variables you can actually use to quantify fitness. And those can be things like growth, reproduction, survival, which we've already talked about. Morphology is another one. These are all, again, these observable physical characteristics. Something like enzyme activity, development rate. All of those are different response variables you could use in a common garden or reciprocal transplant experiment to quantify fitness and see if there are fitness differences between different populations. So again, everything we've talked about so far in this video, these experimental approaches, they're identifying phenotypic evidence for local adaptation. And oftentimes it's a really important first step to figure out if local adaptation is happening at all. So these are two titles of papers that came out of my dissertation. This first one is from 2013, where I did the study I showed in this video with the graphs with the differences in survival, and I concluded that there was phenotypic evidence for local adaptation in these populations. Later on, which is also a paper I've referenced heavily throughout the semester, I did additional genetic studies to find genomic evidence for local adaptation. Ecological divergence is a slightly different term, but it basically means the same thing, local adaptation. And so the phenotype, finding these phenotype differences is often the first step, but then you can study local adaptation further using molecular methods to figure out what the actual genes are causing these differences. And that's what we'll cover in the next video.